is just now getting to talk to you all about some of the projects that she's working on. And in a few weeks, you'll get to see Brendan also. So please, I think it's fair to say there's a lot of discussion. Oh, I turned it on. I can hold it to my mouth. When I was a graduate student, I sang a song with Francis Collins at Cold Spring Harbor. There is no recording, but I will survive. I don't know. I didn't actually know who he was at the time. Being a new grad student, he was just playing the guitar. That's unrelated to the great work that Seema has done, but hopefully everyone can hear us. And she's going to give you a little introduction to who she is also. So I'll just say that we're very lucky to have her here, and hopefully she'll stay with us for a while.
Thank you. That's so nice. Yes. So yes, hi everyone. Thank you for coming to see my talk. I'm Seema. I am a senior research scientist in Dr. Melissa Wilson's lab, the Sex Chromosomes lab. I'm here at the ASU School of Life Sciences in the Center of Evolution and Medicine. And even just like Melissa said, I thought since I joined towards the tail end of the pandemic and I haven't had a chance to like chit chat with you, I thought I'd give a little bit of an introduction on my background, what I've done, where I'm coming from. So I am a computational biologist. This is a picture of me in high school in my very first computer science class. Yes, I was a nerd. So I went on to major in computer science and biology at the University of Rochester because I loved it so much. And biology was my favorite application of computation, and it still is. After college, I went to a graduate program at UCLA, joining the molecular and medical pharmacology department. And this is me learning wet lab. That's what I did. A lot of what I did in graduate school was I had a chance to learn how to do tissue culture and Western blots and a lot of things I didn't quite get a chance to do in undergrad. I got to pick up on it while also honing my skills in algorithm development and data analysis for these sorts of data. And so after kind of outside of my schooling, I've had a chance to do multi-omic research at a variety of institutions. I was a summer intern at Jackson Lab. I was a bioinformatics consultant at John Wayne Cancer Institute studying melanoma and other immune diseases. I was at TGen in the proteomics lab. I was one of the analysts there. I did a project involving neuroblastoma there. And then now here I am at ASU, and I'll tell you a little bit about what I'm doing there. And throughout this whole time, I have done a lot with education because education, I think, is really important. And I have now taught science to toddlers, to college students, and kind of to adults too, actually. So yeah, lots of really cool and fun science I've done. This is a picture of my very first ASU experience. I'm volunteer teaching for the Arizona Cancer and Evolution Center. This is my little daughter right here. And then these are a bunch of kindergarten students, and we're teaching them about blood using a microscope and models and fun stuffies and whatnot. So yeah, so I've done lots and lots of kid science and lots of, grown-up science too. So if ever you guys are teaching anything, want to chat about it, yeah, come talk to me about it. I think it'd be great. So yeah, so this is sort of my vibe. And then as for today, I wrote out a couple of topics for today, but I really want this to be more of a discussion. This is a good time to ask questions for undergrads, especially. So yeah, so please interrupt me at any time. And basically, I wanted to tell you a little bit about the research that I do, and then if we have time at the end, a little bit about some of the education work that I'm doing with Dr. Wilson. The first project I'll talk about is in the pregnancy kind of sphere with sex differences and gene expression in the human placenta. And then I'll transition out of pregnancy and talk about the uterus and brain with regards to ovarian hormones. And then, like I said at the end, the course, if we can. So first project in placenta. So an introduction to the placenta for those that don't know much about it. So the placenta is a specialized organ that forms at the interface between a pregnant mother in her uterus and the growing fetus. It allows for the blood of the infant and the mother to meet without actually mixing together. And there's a lot of interface between to allow all kinds of different exchanges, including methods to deliver oxygen, nutrients, and immune protection to the growing fetus. And then also remove waste from that fetus and filter it through the mother's body. So it's very cool and very interesting for many reasons. But one thing to know for the purposes of my talk is that the placenta and the baby are formed from the fertilized egg. And so the fertilized egg splits off early in development, and it becomes the placenta and the baby. And so the genotype between the placenta and the baby, they match, right? So if you have a female infant, you'll have a female placenta. You'll have the same sex chromosomes, XX in most cases. If you have a male baby, then you'll have a male placenta, XY in most cases. So that's relevant. That's how you can have a male placenta and a female placenta. And the reason I mentioned that is because we're studying sex differences, and chromosomal sex chromosome complement is very important to that, as well as many other things with regard to sex. And so previous research has shown that there are differences in the placenta and the baby based on sex, based on genetic sex. So in females, so when the infant is female, studies have shown that females are smaller on average at birth than males are. And studies have shown that their placentas are more adaptable. The fetal-placental interface is more adaptable to stresses in the mother, environmental stresses, and also have a higher reserve capacity. So more things, stored up for whatever might come its way. And as a result, on average, there's an increased survival when the placenta or when the fetus is female. And in males, on the opposite side, they are on average higher in birth weight, and the placenta has been shown to become less adaptable and has less of a reserve capacity. And so, in the end, there's an increased intrauterine morbidity and mortality rate when the fetus is male. And specifically, one of the complications that we're going to be talking about today is preeclampsia. Preeclampsia is a condition where it's high blood pressure in the mother and proteinuria in the mother. That means there's protein in the urine. That's how preeclampsia is detected, although predominantly it's with the high blood pressure. That's the main component. And that can lead to preterm birth for the baby and then increased risk for organ damage in the mom. And the only surefire way to help someone that has preeclampsia is to deliver the baby early. So that's what the preterm birth is about. And so that's bad for the baby in a lot of different ways. Not enough time for it to develop all its internal organs, immune problems, things like that. And so depending on if you have a female or a male, the there's early onset preeclampsia and late onset preeclampsia, and females and males have different rates and instances of those. With the male, it's more early, and then with the female, it's more commonly in late. And so the idea is to try to understand in this project that I'll be talking about, what are the differences in the placenta based on sex, and then how could those lead to different outcomes in different complications. So in our lab, we've published that there are sex differences in placental gene expression in uncomplicated pregnancies. So taking preeclampsia out of the picture, even when there are no problems in the pregnancy, we have genes that are differentially expressed. And so to explain this, if people haven't seen this type of figure before, this is called a volcano plot. On the x-axis is essentially the difference in average expression, and it's given a sign, positive or negative, based on which samples it's higher or lower in. And then on the y-axis is increasing levels of statistical significance. So the less that these results would be due to statistical chance, right? Just chance. And so practically the way that you can read this is if it's up in this quadrant here, there are genes that are having higher expression, significantly higher expression. And on the side is genes that are higher expression in females. And so all these spots are genes that we're measuring their expression, and we're looking at female versus male. The colors of the points all represent what chromosomes these genes are found on. So the green ones are X-linked, so those are from the X chromosome. The red ones are Y-linked from the Y chromosomes, and the purples are autosomal. And so you can see, so this left-hand side is late first trimester pregnancies. You can see that there is a smattering of both. Though predominantly Y-linked genes in the males, which, to some extent is expected, but you can see that there's a lot of different other kinds of genes also differentially expressed between the males and females. And then when you get to term pregnancy, so that's 36 weeks and beyond, and 40 is, full pregnancy, if you will, then we see almost all sex chromosome genes that are differentially expressed. And so we wanted to compare this scenario to what would happen in a pregnancy complication. And so to do this, we paired up with Valleywise Hospital here in the Phoenix area in Arizona. And we were able to enroll pregnant Hispanic women that they see. And they were in either one of two groups, blood pressure normal, which is normotensive, or elevated blood pressure, which is the hypertensive group. And other than that, they had no significant differences in any other kinds of factors like age or BMI or any other sorts of issues. And then they had no other pregnancy complications like gestational diabetes, for example. They were meant to specifically study this effect. And so we collected with them placenta at full term delivery. And we did two types of sequencing. One is whole exome sequencing, and the other is RNA sequencing. The first is for looking at genetic variations in these placenta, and the second is for gene expression. All of what I'll show today is in the gene expression world. And so total, we were able to collect 20 placental tissues, half from normotensive and half from hypertensive. And we were aiming for a 50-50 split for males and females, because that's what we like to do to study sex differences, right? And in the normotensive, we were able to hit that at 5 and 5. In the hypertensive, we had 4 and 6, pretty close. Yeah. And so for the pilot study, where I'll be showing results today. Yeah, please. No, please don't interrupt. Yes.
It's a complicated structure.
Yes.
Did you self-dissect any particular layer?
Yes.
Like that?
Yes. So we were trying, so there are layers to the placenta, and we were trying to get kind of the inner layer, which is called the villus tissue. And so when you take a placenta out, there's kind of the mom-facing side and the baby-facing side, right? And so we lay the placenta out on the mom-facing side, and then we're taking tissues kind of from the baby-facing side. And then the placenta shape is kind of a circle, like a round, like plate, shape. And they're actually, so the people at the biobanking facility in Valleywise were able to collect samples from multiple quadrants. So they kind of drew a quadrant in the placenta and took 2 samples from each quadrant. So it's very exciting. Yes, we were able to, in the pilot round, just sequence 1 of those samples, but we have more money now, so more samples. Yes, yes, very exciting. But yeah, for now, we're just doing the 1 from each placenta. Yeah, yes, good question. Thank you. Yes. And so these are the results that we found with that first set of pilot results. And so firstly, we found genes that distinguish between sex and hypertensive status. And so basically what I did is I tried to look for genes that distinguish between these 4 groups: female normotensive and hypertensive, male normotensive and hypertensive. So this representation is called the clustering heat map. So on each row is a gene, and they're colored according to their relative expression across all of the samples. So red is high, blue is low. And then the columns are each 1 placenta, and then they're given colors based on their grouping, right? And so if you look for genes that, are there any genes that separate between these 4 groups? The answer is yes. And so when you look at genes that at least distinguish between 2 of those groups, at least 2, maybe more, you can see that the males, which are on this side, so these teal colors are the normotensive samples, these are the hypertensive samples in the male placentas, right? So placenta is connected to a male baby. You can see that there's a lot of difference between those 2 groups, right? There are genes that are high in normotensive that are very low in hypertensive, and the opposite, low in normotensive and high in hypertensive. So they're very, very different from one another. When you get to the female side, however, there isn't as much difference. There are some genes, if you look kind of carefully, you'll find that are kind of more blue on the side, red, and then red and blue. So there are some, but certainly not as dramatic a change as we see in the males. And so if you were to represent this data in a different way, if you just plot if you plot these samples on a map, you can see that the male normotensivs are distinct from the females, and they are themselves distinct from the male hypertensive. So the males are distinct in both definitions, and the females are more alike to one another. Yeah. And so knowing that this is sort of the trend that we see, what we wanted to do is kind of test to make sure that how does our data compare to other preeclampsia data sets that have been studied? And there are a ton of them. And certainly, when you try to get even closer to our data, like the same kind of experiment, RNA sequencing, it gets less and less and less. And actually reported on sex, 0. It's us. So here we are. But yeah, and so we tried to use one recently published data set that just came out in 2023 this year, where they had preeclampsia data and they had the sex of the samples marked. And they had the same kind of experiment we're doing RNA sequencing for gene expression. And so if you take each point here as a gene, and if you plot the fold change, so the difference in average expression just in the males, so if you separate the males from the females, and you look at the fold change in that published data set versus the fold change in our data set from Valleywise, you can see that in the males, they're quite well correlated. And here are the stats and figures. You can see the p-value is 1 to the minus 443, very significant. for sure, not due to statistical chance, right? So in the males, you can see that the data is well correlated. And some of these markers, LEP and FSTL3, they're known markers of preeclampsia. And people haven't previously characterized that only in male preeclampsia or female. They just report on preeclampsia as a whole. But actually, in our data, it indicates that this is from the males. The signal is dominated from the males, and it's something that you wouldn't see unless you break up the samples, and so when you look in just the female comparison, you can see that they're not well correlated at all. And one thing I wanted to mention here, so we're also in collaboration with Flinders University in Adelaide, Australia, where they're testing this on other preeclampsia data sets that they're working with and seeing very similar results. That they're seeing kind of standard preeclampsia markers and a high correlation in the males, but then the females are all over the map, a lot of different things. Did I see hand? Yes, hand. Yes.
Very cool. I mean,
Yeah.
things linked all sorts of issues associated with diabetes. Did you rule out that any of this was gestational diabetes related?
Yes. So in our study, yes, in our study, we didn't enroll mothers that had gestational diabetes, but also a good question because we're trying to apply for a grant and we're going to include diabetes in that. So I'm super glad to hear that you're interested in that kind of thing because we're hoping to find out if that's a factor in things. Yes, please.
What is the comparison within your data for the y-axis there? Like how associated are they? Or the effect of preeclampsia within sexes compared to each other? Like, are they ranked order similar? That sort of thing?
Between each other? Sorry, what do you mean? So meaning are the things that are high?
Right, the hypertension level change within males and then within females, right?
So like, for example, if you had something up here in the males, would it be kind of up here in the females? Is that what you're asking?
Correct.
Yes. And the answer is yes, but at a much, much, much less degree in the females. Much less.
Sizes for rank order change rate relatively.
I think we haven't directly tested it, but we do see that things that are on the high side in the males are also seen in the females at just a much smaller effect. So yeah, especially the ones that we're looking for, like known preeclampsia markers like LEP and FSLP3. They do have some change in the female, but not at all to the same level as what's in the male. Yeah. So we wanted to look and see what these genes do. Looking first in the males, we used a tool called Metascape to look at what kinds of functions are associated with these genes that are changing in the males, and with the intention of trying to figure out, what's going on in the placenta in the males, And you can see in the genes that are downregulated in hypertension in just the male placentas, you can see a lot of different kinds of organization and transport related activities, cell death related activities, responses to hormones and metabolism related activities, and then protein phosphorylation, which has to do with cell signaling, various cell signaling processes that definitely all of these processes are very important for normal cell behavior. It's definitely yes, all interesting genes. So all of these categories that I've listed here, they're enriched based on genes that are found in that list, right? So we can always go into any of these functions and see what genes are giving us that signal and then read about them and learn about them and figure out what the role in these situations are. And so then for genes that are decreased in the hypertensive in male, lots of, again, cell cycle related things, mitosis related, some more metabolism, DNA repair, all things that we would expect to be with normal cell function. And there's also a lot of genes that are involved in mitochondria. And there's a couple of other papers that are starting to talk about mitochondrial function and preeclampsia. So we're hoping that these results will help us to give some molecular underpinnings to those results and understand more about what genes can be involved. And then switching gears to the females, there weren't as many genes in this category, much less. But those genes that were differentially expressed had mostly to do with the spliceosome. So there might be some alternative splicing. So some of our future work will hopefully deal with alternative splicing and see if there's any increase in alternative splicing or decrease in alternative splicing in these results. And then for the females, the genes that are decreased in hypertension had mostly to do with responses to hormones and metabolisms, but several that had to do with the immune system. So that is, the immune system is very important in pregnancy as a whole. And so when we see things regarding the immune system, we always get very excited and try to learn more about that. And that's one thing that we're going to try to do in the future with more of our samples. And to further look at the immune system, we used another method called CyberSort, which is a cellular deconvolution method. So basically what it is a way to take cell types that are available in different tissues. So we've used a published data set of single cell RNA sequencing to look at cell types that are present in the fetus and the fetal placental interface, right? So all kind of the cell types that you would expect to find in the fetus and placenta, right? And so what we can do then is using those signatures, we can mine our data, which is, kind of a smoothie of all the genes, basically. It's a bulk RNA sequencing experiment, so it has all the different things in there. And so we're going to try to, maybe gross. Yeah, she's crinkling her face. I don't know. I think of it as a smoothie. I don't know. I don't know.
Just that it's
Maybe that's gross. Hard to say. It's good that this is before lunch, not after, right? Yes, but no, I think that the cool thing about it is, though, you can get an idea of what kinds of cell signature or cell types can be contributing to this sequencing experiment, which looked across the bulk tissue, say, right? And so using these results, we were able to see a couple of T cell populations, CD8 positive T cells and natural killer T cells that have a difference in the females, but not as much in the males. And so if you look at CD8 positive cytotoxic T cells, from the normotensive to the hypertensive in the females, you can see that it goes from low to higher. And our sample numbers are quite small, 5 and 5, so p-value of 0.1 is kind of on the border of what we might consider to be significant, but you can see visually that, there could be a thing there. Yeah. So there's definitely a difference there. And then certainly it's different from what you see in the male situation, where both the normotensive and the hypertensive are kind of on the same range, right? And then similarly, we see a change in the females and the males for natural killer T cells, because the hypertensive females, there was no immune fraction detected there using this method. And in the males, you can see that there's a range, some high, some low. But yeah, so definitely that could be one of the things in the females that's influencing the gene expression changes that we're seeing. Yes.
Do you see other cell type present just in the portfolio, since Severshore gives you like multiple
Yes.
cell types? Is there other? So I appreciate this one is
Yeah.
different, but are there other cell type differences overall?
A little bit. Yeah, there are some that are a little bit different, but these are the ones that are the most. So it's, I didn't see that much. However, we are working on kind of making, there are multiple different single cell, like 3 kind of good single cell data sets out there. So we're kind of working with our collaborating lab to see if we can work out using multiple data sets and getting in more cell types and try to expand this a little bit more. So these are kind of the first ones that popped out using one single cell data set. But I think combining the results from multiple data sets can give us more cell types. And so we can probably see a better range. And to generally answer your question, we do see some differences, but they weren't that significant. It's kind of not, the effect size is more small. So we weren't sure whether that was variable or not.
I think you're looking at single cell generated from placenta.
Yes.
Right?
Yes.
Specific. And another
Yes.
one that has decidua in it also.
So, well, this one has some decidual tissue as well. Yes. And there's one that's, I'm trying to remember exactly. They all have some amount of maternal tissue, but the amount is kind of different. The stage of pregnancy that they're looking at is different. The reason I chose this one is because it's also taken at term, just like our data was. There's another one, a very famous one from Ventotormo et al. That does it at early pregnancy. So I think we have a little bit of space to grow as far as what we include, but this was our first pass.
And the reason I ask is this is, at least to me, quite provocative because of, as you're probably aware, the first author, Dr. or the senior author, Dr.
Yes.
Wilson, and maybe you're going to get to this, is the pregnancy compensation hypothesis. Difference in the immune system for males and females.
I will leave her to talk about that one day because it's too much to do today, I think. Yes. But yes, I agree. Yes.
Well, along those lines, so this, forgive me if it's way off base, but these are all shared fetal immune cells. They can't be maternal immune cells.
So in the single cell data, they labeled the cells as being fetal or maternal, using their methods to kind of say what kind of a cell it is based on other markers of that cell. And then I'm trying to remember if they had a male fetus, and that also helped them to distinguish which is fetal and which is maternal. But I think So that's how they know it's fetal.
How strong fetal T cell.
Good question.
Like how much maternal nutrition. I'm just curious.
Good question.
The pregnancy compensation is all about the fetal mother and not about the fetus.
Yeah, and we do have a data set in uncomplicated pregnancies where we also collected the decidua, and so that's on my future directions to look more into that question in the decidua as well. But good question. And I can't remember exactly if in this data set they specifically marked CD8 positive T cells from the mother. I can't remember exactly, but that's a good question. I'll look and see. Yeah.
Do you have the mother's genome?
No, not for this one. Nope. Yeah.
Because that, one of the things you could conceivably do is subtract out
Mm-hmm.
the maternal contribution.
That's
to
true.
Do the analysis.
Yeah, but not for this data set. But yeah.
We didn't talk about it much, but we did do analysis for maternal contamination in all of we looked for markers of residual expression and saw none.
Yeah. Low levels.
Yeah.
Nothing.
Yeah.
As close to none as we'd feel comfortable saying is none, right? Yeah.
So I think that at least here, at least in our data, whether it's matching something from fetus or not, I think that this is fetal expression.
Yeah, and generally, I mean, this is cell fractions of immune cells because we're want to talk about immune cells. But if you look at other cell types you would expect to find in the placenta, you would definitely find those cell types. Extravillous trophoblasts, syncytial trophoblasts, all the different cell types that we know are in large numbers. Those are there too. So that makes us feel more comfortable that we're looking at the tissue that we think we're looking at.
I was just quickly Googling because so I work on malaria and placental malaria is a big issue. So malaria parasites invade the placenta.
Oh.
And there's actually a difference between women having a female versus a male fetus, where female fetuses are more likely to get placental malaria than male fetuses.
Oh, dude, what? I wonder. That's so weird. I wonder why.
That is not the act I'm collaborate on, but
Yeah, we should talk after. That's very interesting. Yeah. So next steps, I feel like we've talked about it. We have more samples. Our conclusions are that there's more differential gene expression in male placentas between normotensive and hypertensive. This male signature includes known preeclampsia markers, while the female signature is not quite dominated by those markers, and they're consistent between multiple data sets. The gene expression differences in females may be due to different immune cell proportions in hypertensive pregnancies. And now more regions, and we're also wanting to look at the decidua. So yeah. So switching gears, unless there's questions, thoughts? Okay. But switching gears, I also wanted to tell you about another project we're working on in collaboration with Dr. Heather Bimonti-Nelson and lab here at ASU, where we're studying the effect of ovarian hormone loss on gene expression and cognitive function. An introduction to our happy ovary here and happy uterus. So the uterus and the ovaries are a key part of the female reproductive system. The ovaries, there too, left and right, they contain the oocytes or eggs in females, and then they can travel through the fallopian tubes into the uterus, where in pregnancy, if they're fertilized, they can implant and cause pregnancy and with placenta and fetus, and most of the time, they're shed through the menstrual cycle. And so the ovaries produce a bunch of different kinds of hormones, but the main one that I want to talk about today is estrogen. There are different types of estrogen that are produced over the lifespan of the female. In their childbearing years, there's estradiol. During pregnancy, it's mostly estriol. And then there's another one called estrione that's after menopause. So different kinds of estrogen. Yes.
Estriol is made by the placenta.
Oh, oops, sorry. Yeah, I should have. Oh, yeah, that's right. Yeah, sorry. I don't know why. Yeah. Thank you. Yes. And so the estrogen that's produced by this gland is involved in health and proper function of many different organs in the body, which is not something that people talk about that often, but it is. The one that we're going to be focusing on today more is the brain. There's thought to be a neuroprotective effect of estrogen. And cardiovascular system and skin and bone and urogenital systems are also influenced by estrogen levels. And so sometimes, though, surgical removal of the uterus, which is called a hysterectomy, or surgical or a removal of the ovaries, which is called ovariectomy, is used to treat benign uterine conditions. And so, and as far as so there's that can include abnormal bleeding or fibroids or things of that nature. But the thing is, even though estrogen is involved in a lot of normal function for all kinds of different organs, the full effects, molecular effects downstream of removal of these organs is not well characterized, and more is being learned every day on the effect of estrogen in general, right? And so we're interested in finding the molecular effects downstream of these hormones that are being produced by these organs. Speaking of the neuroprotective effect, also I've gotten some funding from the Arizona Alzheimer's Consortium for this work because there are sex and aging effects on Alzheimer's disease that we think might be involved in some of the stuff that we're studying. The incidence of Alzheimer's disease is nearly twice in females compared to males. Deterioration of cognitive abilities is shown to be far worse in female patients. Females are more vulnerable to genetic risk factors like the ApoE4 allele. And this has caused scientists to form a hypothesis called the estrogen hypothesis, where the loss of the neuroprotective effect of specifically estradiol is thought to further Alzheimer's and other cognitive impairment in females more so than in males. And there have been studies that have shown that there are indeed Alzheimer's biomarkers in the brains of postmenopausal women. So this is all something that people are thinking about and that we are thinking about too. And so in collaboration with Heather Bemonte-Nelson, Dr. Bemonte-Nelson's lab, we're studying a preclinical animal model of reproductive tissue surgery. And so they have a rat model. These rats are at reproductive age, where they have control surgery. So that just means that they do the same kind of anesthesia and kind of open the rats, but don't do anything beyond that, just stitch them back up. And then a surgery called ovex or ovarectomy, where the ovaries are removed. A hysterectomy, where only the uterus, the uterine horn is removed. And then surgery where they remove both, right? And so they've shown that these animal models actually have a cognitive effect that you can study as well. So in this, there are many figures in this paper. It's very cool. But they studied, in this figure, I'm showing the effect that they see on working memory errors. And so in a sham model where you do surgery but don't remove any of the organs, you measure memory. You have this rats learn from a particular activity, and then you test their memory after some amount of time. They took a baseline amount of time for that experiment versus a delay, so that's even more time between the learning phase and the memory phase. And they measured how many errors, that's on the y-axis, how many errors these rats made. And you can see that they're fairly similar, baseline and delay in the sham. But then when you remove the ovaries, there's a significant increase in the number of errors made by those rats. And for me, I don't know if anyone else is shocked by this, but like I had no idea that the reproductive system had anything to do with memory learning. I mean, it was all news to me, and I found it super interesting. So yeah, there was some increase when you looked at the hysterectomy animals. You can see, some, but definitely anything involving the removal of the ovaries, you definitely get a significant change in terms of baseline versus delay. So there's definitely a cognitive difference between when you remove the ovary and when you don't. So taking these animals, we are doing an experiment to measure cognitive effects. And so the way that we did this is we kind of followed this timeline. So the animals arrived to the Bemonte-Nelson lab, and after a week, after they got settled, they did surgery. And so in our case, for this first round of experiment, we're only looking at ovex and sham. We hope to do hysterectomy and ovohys later on. But yeah, and so in this case, they either got ovex surgery or sham surgery. Then they were given several weeks to recover. And then they were subjected to a variety of learning and memory tasks, and they collected the data for all of these. And after these tasks were performed and they had some time to simmer down, the animals were sacrificed, and we collected a number of tissues in these animals. And we also did a vaginal smear to look at their menstrual stage. So we noted that as well in case that comes up later. And so we collected uterine and brain tissue samples, and we studied the gene expression in those samples using RNA sequencing. And then we did differential gene expression analysis, just like we did in the case of the placenta a minute ago. And then we also collected blood samples to look at circulating hormones in these animals. And then, of course, the learning and memory data for cognitive and behavioral analysis. And so this is sort of our grand setup. And today I'm going to be talking only about the uterine results. I'm currently in the process of looking at the brain. I'm very excited. We're all very excited. But yeah, today I wanted to talk about the uterus. And the main take home is that there's a huge difference. There's a huge difference in uterine gene expression after removal of the ovaries. In fact, it's such a cool signal, such a big and deep signal that all the other signals we see seem lame in comparison. So it's changed our view from here on. But yeah, there's a huge signature that we see. And so again, in a heat map, the rows are genes. They're colored high and low, red and blue. And then the columns are uterine samples, uterus samples from each of the different rats. The ovex samples are here on the left in purple, and the sham samples are here on the right in green. And you can see that there's definitely a difference between the two. The groups are organized by similarity to each other. So you can see that the ovex samples group together very tightly, and the sham samples are apart from that. The other thing I noticed right away is that they're very consistent. The signature and the ovex animals are quite consistent across the entire set of ovex animals. There's a set of genes that are definitely turned up, and then a set of genes that are definitely turned down across all the animals. Whereas in the sham, you can see some substructuring, some subgrouping within those, and almost we see kind of a cascade, just visually, which we think is just super cool. So right now we're focusing on the differences between the OVEX and sham groups, but we also want to study more about the shams themselves and see if we can explain some of these differences. Maybe their menstrual stage, maybe there are other features of the rat that call for that. Have these gene expression patterns. But right now we're looking at the difference between the OVEX and the sham, and the number of differentially expressed genes is over 2,000. It's a huge amount of genes that are differentially expressed. And that just shows how closely linked the ovaries and the uterus are. And further, it shows what genes are directly connected in terms of the effect of these ovarian hormones, which are missing in the OVEX, right? Yeah. So again, we wanted to look and see what these genes are doing. And so in the genes that are increased in the OVEX animals, we're looking for, so these are things that we're looking for to relate to uterine function, as well as potentially through, if there are secreted proteins, for example, there are secreted genes that create secreted proteins that might be communicating between the uterus and the brain, that might also have some impact on the brain. But we're basically looking at ways that uterine function can be changed in the absence of the ovaries. And so we see a variety of signaling pathways, and so we notice, synapse, nervous system development, brain development, neuron projection. And so even though this is the uterus tissue, right? So this would be neurons in the uterus, right? Not neurons in the brain, which is future talk, right? It does make it interesting to us that there are neural pathways that are affected by ovarian hormones. Right, so this is stuff that we want to learn about and see what effect the ovaries have on the uterus, and then how that might, at some point, systemically cause a difference in the entire animal. And so we also see a variety of transport, ion transport channels, responses to hormones, a bunch of different secreted factors, growth factor responses, a lot of really interesting aspects that might explain how the uterus would be performing differently in the absence of the ovaries. And then on the other side of genes that are decreased in the OVEX uterus, a lot of cell cycle related and replication related pathways, different kinds of reorganization pathways. So this might explain structural changes you see in the uterus, because the uterus kind of shrinks way down. Shrinks way, way down. So there's definitely physical changes that happen in the uterus after removal of the ovaries. We see various hemostasis, so kind of balancing out factors, trying to keep the function of the uterus, but can't in this different case. We also see a little bit for immune response, it's innate immune response, inflammatory response. So there might be some response of the body detecting that something is wrong, potentially. So again, all of these are super interesting to us and our various collaborators, including Dr. Jason Newburn in the back. And so we're looking at the huge gene expression signature and trying to figure out what it's doing and mining the data, which is kind of our bread and butter. Yeah.
I'm curious, did you do this class of analysis, not segregating up and down, just like genes that are different? Do you get different pathways out of it?
Not as such. I mean, we have done that, and you see kind of a mixture of the two, kind of largely speaking similar categories of things. But you're right, though, that would be good to look at, because there might be pathways where some things are up and some things are down. So yes. Yeah, I don't know. Yeah, but basically though, so we're in the process of understanding this uterine data, and we're all very super excited about the brain data. So in the brain, we collected actually four regions of the brain involved in learning and memory from the same animals. So the cool thing about working in an animal model is you can have very good controls, right? In humans, not so much. But in animals, you can do so they're the exact same brain, or the exact same animals. We collected the uterus and the brain. And so we are looking to see molecular underpinnings that are responsible for cognitive changes in these animals due to ovarian hormone loss. And we're also going to be integrating this with human data that's available. So both from normal aging, so normal females across the lifespan, including transitions to menopause, as well as specifically about Alzheimer's disease. As you can imagine, there's a lot of human data out there, but always very interesting to use other types of data to understand that signal better. So yeah. So I have a little bit of time. You guys still doing okay? A lot of things I'm throwing at you. Yeah. Okay. Cool. Because I just wanted to quickly tell you guys about some teaching that we're doing together. It's very cool. I'm very excited about it. So so Dr. Melissa Wilson and I are leading the teaching of online genomics research courses here at ASU Online. And so these are in the format of what's called a CURE, a course-based undergraduate research experience, right? So basically these are courses where the students are learning and we're guiding them through doing a research project. Yeah. So the format of how the course is taught is Dr. Wilson teaches the first session in the fall semester. It's a prerequisite computational genomics course to teach them general things like, how do I log on to the ASU network? And how do I look at quality control measures? And what is sequencing? And things like that, right? Just general kind of thing. Yeah. And then we take over together and teach the research portion of it right after that in the B session. And currently we have funding for 3 rounds and we've completed 1 and 1 is about to happen. Last year, the pilot online genomics cure had 13 students. Fabulous. And then 2033, we currently have 50 people. So huge change. We're happy/nervous/happy about it. It's going to be interesting. Yeah. So the course development is some of what I'm going to tell you about today, how we come up with this course, how we teach the course, how it works. Because again, this is an online situation and this is taught asynchronously. So we have had students from all around the world. We've had students that have families, that have jobs. We had a student that's a medical doctor and we didn't even know until the course was done. He's a pediatric oncologist? Hematologist. Sorry, hematologist. No, like no big deal, just anyway, yes, very diverse and amazing students that wouldn't normally get a chance maybe to take this opportunity, to do research otherwise because they're busy with life. So every time we come up with a cure, we think of a novel research project for these students to do. And I think we've sort of figured that the main thing is that we have to have analysis that can be split among the students, essentially. That's kind of the theme between the projects, I would say, because the projects that we're doing this year and last year and this year are quite different from one another. But that's something that they have in common, that it's analysis you can split. And like I said, it's conducted asynchronously online, so they can open up their learning materials whenever they want. They have assignments that they're doing, but we have lots of ways to kind of have them get help from each other, and I'll talk about that in a second. And then for each project, we create custom learning materials and assessments so that we can make sure that they're doing okay and learning what we want them to learn. Yeah, super exciting. And so last year, I thought, since I'm speaking with scientists, I want to tell you a little bit about the question that we did last year. We wanted to test the robustness of our placental sex differential gene expression results, which part of which I put in my first part of the talk. And we wanted to see how robust those were to different data processing parameters. In our case, we looked at trimming. And the part of this, for those that are kind of new to research, younger students, is, we have a sort of pathway that we take through the data, processing steps that we do in the lab that we think make sense. But generally speaking, we don't have time to test a lot of these things. We kind of, I join a lab and then someone else has done this thing and then I just keep doing that thing unless there's a problem where I have to change it. But then really so it's kind of done that way. But in this course, we wanted to teach the kids about, how data is produced, how it's processed, and what impacts that can have, if any. Right. And so one thing that we always point out to the students when we teach them is that since this is research, we don't know what's going to happen. It could be that we go through all of this and see no difference. It could be that we go through this and see a huge difference that we should have known when we published, but we just didn't think to test it, right? We don't know. It could be anything on that range. And that's something that's very difficult in a good way for the students, because that's what research is. And, that's what you should learn. You should be okay with whatever happens. You need to follow the results, right? And so what we did is we took our placenta gene expression data. We just took the first batch of our first uncomplicated pregnancy data, so we didn't have to deal with batch correction and other kinds of things. And we took that data just as it was in an untrimmed scenario. And then we created different versions of that data where we had varying trimming parameters. So we did the same trimming software that we did in the original paper where we published this work. And then the 2 parameters were min length, which is the sequencing quality trimming score. That was one. It's called TrimQ in BBDuck, which is the software that we used. And then min length, which is the portion of the sequencing read remaining after you trim away all the low quality sequences. So we varied those and had a range of 9 trimmed data sets. So we varied each of those parameters along the spectrum. We did a literature search before the kids started to see what kinds of parameters make sense based on what other people report in papers. So we tried to set up the project as best we could. And develop learning materials where we talked to them about all this stuff. I put a little video of myself on the course saying, hey, this is what we did before you got here. Yeah. And then we had the students in class do differential gene expression using the same tools that we did for the paper. And so we started all together as a class doing the untrimmed case, which we thought of as a control. And then we divided the rest of the trimming cases up between the students. So we had 9 data sets, so 9 sets of students, and I think we had them in pairs. So pairs of students did the same parameters. So that way they had a buddy if they wanted to ask, hey, is this what you got? Right? Yeah. So we divided it among all the students. And then as a group, we taught them how to do research. We showed them how to visualize the sex differential gene expression, how to interpret the results that you see, and how to present those results like within the class to your, the students, and then even beyond in some cases. And at the end, our final project is they have to write kind of a manuscript style report, right? To get them learning about papers, Yeah. And so for the way that we did this course in general is we had 7.5 modules, or each week there was one module, and we had a novel research question kind of inside the world of the cure that we're teaching. For each week, we divided up the learning materials into a biology section where they learned about the background of the research project and other things that they need to know in order to interpret the results. We had a coding section where we taught them, how to do all these things, right? A lot of them it was their first time working in R, for example, right? So we taught them how to do coding. We also developed some template code to try to get some of the people kind of all on the same level, because we had some students that had programmed before, some that had never programmed before really, except for what they saw in the prereq, so we had a whole range, Yeah, so we tried to get them a little bit on the same level by giving them a little bit to start with, and then had them, make changes from there. And then we also had a professional development section each week where we taught them how to troubleshoot, how to Google for solutions on the internet, what is a paper? How do papers get reviewed? All of these sorts of concepts that we use, in an active research environment. And we wanted the students to get, real instruction from people that do it every day. So yeah, so that was really, I think, very excellent. And so for each module also we so I'll also talk a little bit about the assessments and communication. So the way that we tested to see if we did an okay job is we had a learning assessment that directly matched the content of the course. And we did the exact same one at the beginning before they started and at the end after they've had the course. And so that way we get a clear measure of whether they have learned anything or not and how. Right. And if they didn't learn in certain cases, where did they make mistakes? Right. So we leaned on that heavily for the analysis of the course. And by the way, that was featured in a master's project. So we had a student that was dedicated doing that work. She did a great job. And then the other thing that we used a lot in a course is a weekly progress report. So we had this idea that since in Dr. Wilson's lab, we all have to do a weekly progress report, we should have our students do the same thing. So we set up kind of a template for them to tell us about their challenges, how they address those challenges, what they are going to, the results that they've gotten that week, things like that. We had a kind of template that they could follow, and we used those as a main part of their grade, and we emphasized to them that this is about doing the research, not about what the results are. Because again, we don't know what the results are, right? So it's about how you're doing the work and you're trying to move the research forward, whatever the results are, right? And what specifically, because each person is different, what challenges did you have, right? And then we analyze these weekly progress reports, and we use those to figure out different coping mechanisms that the students used, what concepts were difficult for them, how they addressed those concepts. And we used a lot of those comments to make improvements to our current cure for this year. So it's a kind of an iterative process for us too, So it's really awesome, and a lot of great things came from the students. And then we also had asynchronous and synchronous communication throughout the course. And so that included a weekly lab meeting, again, just like regular lab. We had kind of office hours, shared research hours where the students come in, ask us questions. And then we also had a very active Slack channel that we asked them to use. And we actually saw that the students that were engaged on Slack had more learning gains than the students that didn't. So that was pretty cool too. So we had a lot of different asynchronous and synchronous methods for students to jump in if they wanted to. And try to really make it a communicative course. And success, yay. So yes, we're excited to see that in our pilot, something happened, it was good. And so in our pre and post assessments, so again, these are identical learning assessments that were modeled after the materials in the course, we saw a change, and the p-value is significant, 0.003, good times. There's a statistic that kind of gives you an idea of how, kind of what the sense of the effect of this is. This was a medium to large effect. Excited. Yes. And so you can see that most students, it's hard to see, sorry. You can maybe see some of these lines. I learned today that gray is hard to see. good to know. Yeah, but a lot of these lines, which are each individual student, they're going up from the pre to the post, right? So a lot of the students definitely perform better on their second round of the exam. Most of the improvement was seen in the biology and coding sections, because we kind of think that maybe they had some idea of research before they started, and so that kind of changed a little bit. Whereas the biology was quite new to them. Most of them didn't know anything about the placenta. And the coding, of course, is always new to everybody as we go, right? So yeah, 9 out of the 13 students asked to continue working on this project after the cure. So that's good reviews, right? And then 2 of the students presented a poster for undergraduate research symposiums after that in the next semester. And 2 of them, they also got an award. Were one of the top bioinformatics presentations. So proud mama, very nice. Yeah, and a lot of them, we talked to them after they're going to graduate school, they're doing all kinds of things, and we think we may recruit a graduate student to our lab. So fun times. Yeah, so she's great. And so we think that not only was this helpful for the students to see a guided, way to go through research and hear from experts in the field, but also it helped us too, So if you are interested in finding kind of a formal way of training future students and are interested in drawing, especially from the online community, there aren't that many opportunities like this out there for them. It's a wonderful way of increasing the access to education to a crowd that might not have it. And it's a great way to just work with new students and teach them things. It's fantastic. Yeah. So just putting a plug in for future cures if anyone wants to jump on board, right? Yeah. And then I think I'll stop there. I feel like, good, I did it. And so thank you to all the people that worked on all of these projects I'm talking about. Of course, our lovely Dr. Wilson and everyone in our sex chromosomes lab for all their feedback. We work with Valley West Hospital and Flinders University with specifically these lovely folks to do the placenta work. Dr. Heather Bramante Nelson in lab, especially her student Cameron Lizick, and Dr. Jason Newburn in the back for the rat work for ovarian hormones. And then there's a whole team of people to work on the cures and develop all the stuff. And then funding, of course, can't do anything without money. Yeah, so that's all I have. Yes, thank you all for your attention as well. And and questions. I think there's 6 minutes before time's up. So, yes, please. Yes.
Yeah, so people were
Oh, thank you.
the rat model work.
Mhm.
Are you guys also measuring estrogen receptor densities in the brain or in the
Mm.
your
Densities, not exactly, but we have gene expression for the receptor, so we can at least see if there may be in there. Maybe.
Can we do that on both tissue?
Yeah.
probably I don't know for sure.
Do you
Yeah.
have tissue usually?
I usually do a live tissue, you stand it,
Oh, okay.
but you might be able to do it frozen, depending on how it's frozen.
Hmm.
You didn't, but we have frozen tissue.
Yes, fully frozen. Okay, this is-
I think it's frozen.
I-
I don't think it's dissociated. I think it's flash frozen and that's it. I think they just put it.
Good animals that would work.
Hmm.
Yes.
Okay.
Actually, those kind of interesting to see what the percent that's a little bit for
Hmm.
because individuals are going to vary in overall estrogen exposure and
Yeah.
during puberty,
Mhm.
lay down on the receptors, and so individuals with lower levels of estrogen beforehand
Hmm.
or recognize
Hmm.
might have less of a change than the ones who had
Yeah.
higher receptor densities to get
Mhm.
and higher levels of estrogen to get with.
Do you expect
Excellent
that in
one.
the rat model that we thought from all or the same age?
I don't know too much about rats. I most
Yeah.
but in humans we see pretty huge
It
differences.
is,
Yeah.
yeah, I did, not that we shouldn't do it,
Mhm.
maybe we could add that as an investigation where we give them different exposures
Mm.
to see if.
Yeah.
Uh,
Our expression,
Yeah.
expression of the receptors extension correlate with
Yeah. Yeah.
some of the
Yeah. And we can look at differential expression of estrogen receptor targets. So that can also be kind of a stand-in for the receptor levels as far as their density. Yeah. Excellent suggestion. Thank you. Yeah. Yeah. Please.
follow up on that. But estrogen is locally produced in the brain as well. So even if you overoptimize animals, you still
Mhm.
have expression from the adrenals and from local regions in the brain.
Hmm.
So you might want to consider that when you're thinking about
Yeah.
also the different hormones. So you can measure
Mhm.
aromatase expression in the brain, for instance, which would
Hmm.
give you sort of like a loose measurement of how often it's being produced from testosterone in the brain.
Gotcha. Yeah, that makes sense.
Particular is that we do
Yeah.
not see a big signal of
Yeah.
overreactivity on brains.
We do see some,
So it's not that.
but it's not, I mean, it's, like I said, our view is always messed up now because we've seen this ridiculously large signal that we cannot unsee now. So yeah, but it's a much milder effects in the brain.
But for many days, probably.
Yeah, and expected, right? Yeah, I mean, yeah, it might not be, but that's excellent suggestion though. We are focused on ovarian hormones, estrogen, but you're right that it can be other places for other reasons. So that's a good point.
Yeah, if you look at the web, for instance, like they often like locally produced in the areas that give like memory, motivation, things like that.
Mhm. Yeah, and I think one other direction that I didn't spend as much time talking about because it's not as much in my field is we're going to correlate between the learning and memory results too. So, in animals that have more learning errors, for example, then look to see what's higher in those animals within that range that, we have a somewhat small number of animals, but the data is really clean. So maybe we can do some correlations and find some things that way too, of course. Yeah. Any other thoughts? Yes, please.
Probably a very naive question.
That's fine.
I'm from psychology.
Oh.
This is very different.
Yeah.
For me, I was wondering if in humans, if birth control could potentially affect some of this and how genes are expressed. Maybe not living uterus, but how that could potentially affect expression.
I feel like the answer is yes, but I don't know as much about why. I know that there are links between contraception and cardiovascular related functions. So certainly you can have effects outside of the reproductive system. And I would say a little bit yes, but I don't know offhand. What was your name again? I might Savannah. Okay, I'll try. Yes, if you email me afterward, I'll look it up and we can learn together because I don't know, but I feel like a little bit yes. Yeah. Questions?
I think just to add a little.
Yeah,
to
please.
Estrogen definitely was something worth considering. In that data, found a number of secreted molecules. I don't think people have any clue what they're doing. And this idea of the uterus perhaps having more signaling roles than previously appreciated. Really
Yeah, that's one thing I should have emphasized that I didn't. Yeah, so there's this. The uterus is known for pregnancy. It's like it's claim to fame. Like people know uterus, pregnancy, good times. But then with non-pregnant uterus, a lot more shade throwing at, people don't think about the non-pregnant uterus. Some people have called it dormant. Some people have called it useless. Don't approve of any of that because there's a lot of different things going on with these organs that we don't necessarily know as much about. In some cases, these organs are removed without giving as much thought as you should give to that procedure. There are medical instances where you should remove them, of course, but in some cases they're removed and people don't take the time to think about what other effects or give the patients as much support as they might need for other effects that you should look at. Just the more that we know about removing these organs and the role that these organs play in the healthy and in the unhealthy body, the more we can give specialized treatment for these conditions and more we can know about the human body, which I think is really important. And it's funny because I think I'm at the interface of a lot of things that people don't care as much about. Yes. Like I am a woman in science, less. Then I'm a woman studying women. Sex chromosomes are straight up ignored in all the things, less. Yeah. So here I am doing the things. Yes. So yeah.
Great thoughts.
Oh, yes, thank you. Any other questions? Yes, I have my email on thing. Please, questions, comments, concerns, things you think of. Malaria? What? Yes, I have to learn. Yes, but anything you think of, I'm around. Yeah.